Protoplast fusion. Go for protoplast fusion. Protoplast fusion is also called as a somatic hybridization. Also called what's a somatic hybridization. Also called as somatic hybridization or parasexual hybridization. Also called as what? Parasexual hybridization. Parasexual hybridization. Is this statement okay? Clear? Right. Now listen here. From plants, plant cells are isolated. From plants, what are isolated? Plant cells are isolated. Sir, plant cell contains cell wall, yes or no? This cell wall prevents the fusion. What? Cell wall prevents the fusion of these two isolated cells. Prevents the fusion of the two isolated cells. It means the cell wall acts as a physical barrier. What acts as a physical barrier? Cell wall acts as a physical barrier. It is preventing the fusion of two isolated cells. So, if we want to fuse the two isolated cells, we have to remove what? Cell wall. We have to remove the cell wall. Now, listen this statement. Sir, two plant cells isolated from different plant varieties. Different plant varieties cannot be fused. Why, sir? As a cell wall acts as a what? Physical barrier. Cell wall acts as a physical barrier, preventing the fusion of two isolated plant cells. Sir, from different plant varieties, two plant cells are isolated, sir. These two isolated plant cells, we cannot fuse them. Why, sir? Because cell wall, it prevents the fusion of two isolated cells. It means cell wall acts as a physical barrier. So what should be removed, sir? Cell wall should be removed. So cell wall should be removed. Sir, what should be removed, sir? Cell wall should be removed. To remove cell wall, we are having two methods. Therefore, cell wall can be removed by how many methods, sir? Two methods. Cell wall can be removed by two methods. Cell wall removed by how many methods? Two methods. Sir, what are the two methods by which cell wall is removed, sir? Number one, mechanical methods. Number two, enzymatic methods. Mechanical methods, and second one is enzymatic methods. By these two methods we can remove the what, sir? Cell wall. Sir, these mechanical methods are old methods, whereas enzymatic methods are new methods. Go for the first methods. What they are? Mechanical methods. Go for mechanical methods. Mechanical methods. Sir, in mechanical methods, right, in mechanical methods, right, how cell wall is removed, sir? Take a plant cell. Which cell we are taking? Plant cell. Sir, in plant cell, what is the outermost layer, sir? Cell wall. What is the outermost layer, sir? Cell wall. Yellow layer, what I drawn, it indicates cell wall. Whereas this white one, what I am drawing now, it indicates plasma membrane. What it indicates? Plasma membrane. Therefore, this is a cell wall. This white one is a plasma membrane, whereas this is a nucleus, right? This is a cytoplasm. Is it okay? This dots indicates what? Cytoplasm. Whereas this ball indicates what? Nucleus. Whereas this yellow layer, it indicates cell wall. This white layer, it indicates plasma membrane. Sir, this plasma membrane, cytoplasm and nucleus, can we call these three together as protoplast? Yes. Therefore, what comes under protoplast? I am speaking about protoplast, not protoplasm. Plasma membrane, cytoplasm, nucleus, they comes under what, sir? Protoplast. They comes under protoplast. Now, is this statements okay? Right. Now, sir, this is which cell, sir? This is a plant cell. This is a plant cell. Is it okay? Right. Now, what I am doing means I am placing this plant cell in hypertonic solution. Placing plant cell in which solution, sir? Hypertonic solution. Placing plant cell in which solution? Hypertonic solution. Is it okay? Right. Sir, when plant cell is placed in hypertonic solution, when plant cell is placed in hypertonic solution, what happens, sir? Water present in the plant cell comes out. What comes out? Water present in the plant cell comes out. Sir, water came out of the cell. Therefore, this is exoosmosis. Name itself indicates exo means outside. What came out? Water came out. From where? Plant cell. When it is placed in hypertonic solution. Therefore, when a plant cell is placed in a hypertonic solution, water comes out of the plant cell. As water came out of the plant cell, we are calling it as what, sir? Exoosmosis. We are calling it as what? Exoosmosis. What we call it as? Exoosmosis. Sir, due to exoosmosis, what happens, sir? Due to exoosmosis, what happens? Now, see the diagram carefully. In this diagram, plasma membrane is closely attached to cell wall. Yes or no? But as this cell has lost water now, what happens means the protoplast contracts. What contracts? The protoplast contracts. When protoplast contracts, what happens means plasma membrane gets separated from cell wall. What gets separated from what? Plasma membrane gets separated from cell wall. Therefore, plasma membrane has separated from cell wall. I think you are getting the difference in these two diagrams. Here, plasma membrane is closely attached to cell wall. But when water came out of the cell, the protoplast has contracted. Due to contraction of protoplast, plasma membrane separated from cell wall. Plasma membrane separated from cell wall. Plasma membrane separated from cell wall. What separated? Plasma membrane separated from cell wall. Is it okay? It means now this cell is called as a plasmolized cell because it had undergone what, sir? Because it has undergone plasmolysis. It has undergone what? Plasmolysis. Therefore, what happened here? Protoplast contracts. Sir, if protoplast contracts, what happens? Plasma membrane separated from where? Separated from cell wall. It means the cell has undergone what, sir? The cell has undergone plasmolysis. The cell has undergone what? Plasmolysis. Now you tell me, sir, now cell wall and plasma membrane both are separated from one another, yes or no? With the help of a knife, with the help of a knife, now you remove the cell wall. With the help of a knife, we are removing the cell wall. This is the mechanical method what we are using to remove the cell wall. Therefore, with the help of what? With the help of a knife. Therefore, with a knife. With what, sir? Knife. What is removed? Cell wall removed. Sir, if cell wall is removed, what is left? If cell wall is removed, what is left? If cell wall is removed, what left? What left? Plasma membrane, cytoplasm and a nucleus. Yes or no? If cell wall is removed, what is left? Plasma membrane, cytoplasm, nucleus. But these three together called as what? Protoplast. Therefore, what we got? Protoplast. We got protoplast. Is these statements okay? Is these statements clear? Therefore, with the help of knife, what we removed? Cell wall. After removing cell wall, what left? Protoplast. What left? Protoplast. Now, listen this statement. Sir, take a plant cell. Plant cell means having how many parts? A plant cell is with two parts. What and what, sir? Cell wall and protoplast. Now, from this plant cell, what we removed, sir? From this plant cell, we removed what, sir? Cell wall. From plant cell, if cell wall is removed, what is left? Protoplast is left. Protoplast. We are calling this protoplast as a naked protoplast or naked plant cell. Naked protoplast or naked plant cell. Is this statement okay? Therefore, when a plant cell is naked, if cell wall is removed, who is naked? Plant cell is naked. If what is removed? Cell wall is removed. Flower is naked if perianth lobes are absent. Flower is naked if perianth lobes are absent. Yes or no? Ovules are naked if ovary wall is absent. Seeds are naked if fruit wall is absent. Is it okay? Bulb is naked if tunic is absent. These statements we read in first year. Yes or no? When is bulb called as a naked bulb? If tunic is absent, bulb is naked. When ovules are naked, sir? If ovary is absent, ovary wall is absent. When seeds are naked, sir? If fruit wall is absent, if fruit is absent. Sir, when flowers are naked, if perianth lobes are absent, that is calyx and corolla are absent. When a plant cell is naked, sir? If cell wall is absent. Therefore, from a plant cell, if cell wall is removed, from plant cell, if cell wall is removed, right, what is left, sir? Protoplast. We are calling that protoplast as what, sir? Naked protoplast or a naked plant cell. Is these statements okay? Right. This is concerned to which method, sir? Mechanical methods. Now go for number 2. Which methods, sir? Enzymatic methods. Which methods? Enzymatic methods. Go for the second method, right? Enzymatic. Enzymatic method. Name itself indicates enzymatic method, right? We are using cell wall digesting enzymes. Cell wall digesting enzymes. Which enzymes? Cell wall digesting enzymes. Sir, we can call the cell wall digesting enzymes even as a cell wall hydrolyzing enzymes. Cell wall hydrolyzing enzymes such as what, sir? Cellulase and pectinase. What and what? Cellulase and pectinase. Sir, cellulase acts on what, sir? Cellulose. Whereas pectinase, it acts on what, sir? Pectin. It acts on pectin. Is this statements okay? Right? Therefore, sir, enzymatic method. Here what we are using, cell wall digesting enzymes or cell wall hydrolyzing enzymes such as cellulase and pectinase. This cellulase digests the cellulose, whereas pectinase digests what, sir? Pectin. Pectinase digests pectin. Is this statements okay? Right. Now listen here. So till now we removed cell wall from a plant cell. Removed cell wall from a plant cell. Is it okay? Right. From two different plant varieties. From two different plant varieties. What are the two different plant varieties, sir? First one I am taking as a parent one, that is P1. Second one as P2, that is parent 2. These are two different plant varieties. From P1, a plant cell is isolated. Plant cell isolated. Is it okay? From P2 also, what is isolated, sir? Plant cell is isolated. Is it okay? Right. Now, from this plant cell, what we removed, sir? Cell wall. Cell wall removed. What is removed, sir? Cell wall is removed. If cell wall is removed, what we got? We got a naked protoplast. Naked protoplast. Naked protoplast. Sir, here also what is removed, sir? Cell wall is removed, right? Sir, if cell wall is removed, here also what we got? Naked protoplast. We got naked protoplast. Naked protoplast. Is it okay? Right. Now what we have to do, sir? Now we have to fuse this protoplast with this protoplast. Sir, how we can fuse these two naked protoplast with one another, sir? Fusion of naked protoplast. Fusion of naked protoplast takes place by two methods. Takes place by how many methods? Two methods. First one is a spontaneous fusion. Second one is induced fusion. For the first one, spontaneous fusion. Second one is a induced fusion. Isolated protoplast has to be fused. Yes or no? Right? Fusion of isolated protoplast. Fusion of isolated protoplast. Fusion of isolated protoplast which are naked. How, sir? By two methods. By how many methods, sir? Two methods. Right? What are the two methods, sir? First one is spontaneous fusion. What is the first method? Spontaneous fusion. Spontaneous fusion. And the second method is a chemofusion. Induced fusion. Better to call it as induced fusion. What it is? Induced fusion. Okay, what are the first ones, sir? Spontaneous fusion. Now you tell me. From a plant cell, to remove cell wall, we used cell wall digesting enzymes. Yes or no? To remove what's our cell wall from a plant cell, we used enzymes. Sir, when we used enzymes to remove what? Cell wall from a plant cell, then obtained protoplast, that is a naked protoplast, spontaneously fuses with one another. Spontaneously fuses with one another. Is this statement okay? Producing a multinucleated structure. Producing a multinucleated structure. Is this statement okay? Right? Therefore, which fusions are this? Is spontaneous fusion. Spontaneous fusion. Right? When cell wall of two plant cells of same plant are removed by enzymes. By what, sir? Enzymes. Then simultaneously, then spontaneously, simultaneously, then spontaneously, the naked protoplast, naked what, sir? Protoplast gets fused to form a multinucleated structure. To form a multinucleated structure called as homokaryon. What it is called as? Homokaryon. Is this statement okay? Is this statement clear? Now see this statement carefully. Sir, here we are taking two plant cells of the same plant. We are removing the cell wall. When you are removing the cell wall by using enzymes, spontaneously the naked protoplast of the two plant cells gets fused. Is it okay? Right? What gets fused, sir? Spontaneously the naked protoplast gets fused. When this protoplast fuses with one another, what is formed, sir? Multinucleated structure is formed. What is that multinucleated structure called? As a homokaryon. What is that multinucleated structure called? As a homokaryon. Is it okay? Right. Now go for number 2. What is the second one, sir? Induced fusion. What it is? Induced fusion. It means artificially we are making the two naked protoplast to fuse with one another. Artificially we are making the two naked protoplast to fuse with one another. Sir, how we can make how we can carry out this induced fusion, sir? This induced fusion is of two types. Induced fusion is of how many types? Two types. Which fusion? Induced fusion is of how many types? Two types. First one is electrofusion. Electrofusion. Second one is chemofusion. What it is? Chemofusion. Right? Go for the first one. What it is, sir? Electrofusion. Name itself indicates electricity. Yes or no? Right. Therefore, what it is, sir? Electrofusion. Sir, here what we are using? High frequency alternating high frequency alternating electric field with short current pulses, with short current impulses. Is this statement okay? Therefore, sir, electrofusion. Sir, by using high frequency alternating electric field with short current impulses, we are fusing these two naked protoplasts with one another. In the case of what? Electrofusion. Now go for the second one. What it is? Chemofusion. Name itself indicates. To fuse the naked protoplast with one another, we are using what? Chemicals. Therefore, it is a chemical mediated fusion of naked protoplast. Chemical mediated fusion of naked protoplast. What it is called as chemofusion. Is it okay? Right? Now listen here. Sir, during chemofusion, what we are using, sir? Chemicals. What are the chemicals, sir? First one is polyethylene glycol. In shortcut, we are going to call it as PEG. P-E-G. Polyethylene glycol. Second one is sodium nitrate. What it is? Sodium nitrate. Sodium nitrate. NaNO3. And third one is calcium at high pH. We are using calcium when pH is high. Therefore, calcium at what, sir? High pH. Calcium at high pH. Is this statements okay? Right? Therefore, listen here. Chemofusion. By using chemicals, we are fusing the naked protoplast. What are the chemicals, sir? Polyethylene glycol, sodium nitrate, and third one is calcium. But when we are using calcium, pH should be high. What should be high, sir? pH should be high. Is this statement clear? Now listen this statement. Sir, this polyethylene glycol, what it is? Polyethylene glycol is used even to transfer genes during genetic engineering process. Therefore, transferring genes by using polyethylene glycol, which is a chemical, is named as chemical mediated gene transfer. What, sir? Chemical mediated gene transfer. This statement you will get in detail during biotechnology, in chapter biotechnology. Is it okay? Right? We are discussing, right, about polyethylene glycol, right? Therefore, polyethylene glycol. Polyethylene glycol. Sir, what are the uses of this polyethylene glycol, sir? Number one, it is used for fusion, right? It is used for fusion. Fusion of two naked protoplast. Naked protoplast when during somatic hybridization. During which process? Somatic hybridization process, two naked protoplasts are fused by using PEG, right? Next, this PEG also used for what, sir? Gene transfer. Used for what? Gene transfer when during genetic engineering process. Sir, what is another name for genetic engineering process? Another name for genetic engineering process. Another name for genetic engineering process. rDNA technology. Sir, using a PEG for what? For transferring genes when during genetic engineering process. Can we call it as chemical mediated gene transfer? Yes. Because by using chemical, which chemical? PEG, gene transfer has taken place. Therefore, using PEG to transfer gene, it is a chemical mediated gene transfer. It is a chemical mediated gene transfer. It is a chemical mediated gene transfer. Is this statement okay? Right. Now listen this statement. Sir, naked protoplast fused with this naked protoplast. Any substance which is involved in the fusion of these two naked protoplasts, we are calling them as a fusogen. What they are called as, sir? Fusogen. Fusogen. What they are called as a fusogen. Therefore, what is the meaning of fusogen, sir? Fusogen means nothing but the substances which are involved in the fusion of these two naked protoplasts. Substances involved in the fusion of these two naked protoplasts, we are calling them as what, sir? Fusogen. Is it okay? Right. Sir, when these two naked protoplasts fuses with one another, then what is formed, sir? Hybrid protoplast. What is obtained, sir? Hybrid protoplast is obtained. Hybrid protoplast is obtained. Now what we have to do? Now you culture this hybrid protoplast on nutrient medium. On culture. On what? Culture. On culture medium. On culture medium. What this hybrid protoplast will do? This hybrid protoplast develops a cell wall. What? Hybrid protoplast develops what? Cell wall. Divides continuously. Who? After developing cell wall, this hybrid protoplast divides continuously and produces somatic hybrid. Produces a new plant called as a somatic hybrid. Produces a new plant called as somatic hybrid. Is this statement okay? Sir, this is hybrid protoplast. Now we are growing this hybrid protoplast on culture medium. Is it okay? Therefore, on culture medium, write on culture medium, it means hybrid protoplast develops what? Cell wall. Develops what? Cell wall, and it divides continuously. Divides continuously and produces what, sir? A new plant. Produces a new plant. What is that new plant called as a somatic hybrid? What is that new plant called as a somatic hybrid? That new plant is called as a somatic hybrid. Is these statements okay? This is which process? Somatic hybridization process, also called as protoplast fusion or parasexual hybridization. Is these statements clear? First we removed cell wall. We got naked protoplast. By using fusogen, right, we are fusing these two naked protoplast. Then what we got? Hybrid protoplast. We are culturing this hybrid protoplast on nutrient medium. Then hybrid protoplast developed cell wall. Developed cell wall. Okay. Divided continuously and produced a somatic hybrid. Produced which hybrid, sir? Somatic hybrid. Is this statements okay? Now listen this statement carefully. Sir, here we are fusing protoplast with another protoplast. Yes or no? Sir, what is the meaning of protoplast, cytoplasm and nucleus? It means the genes present in cytoplasm fused with the genes present in the cytoplasm of this. Yes or no? Right. Similarly, right, nuclear genes present in nucleus of this fused with genes present in the nucleus of this. Therefore, here fusion of cytoplasmic genome and nuclear genome has taken place. What and what? Fusion of cytoplasmic genomes and nuclear genomes has taken place. Is this statement clear? Okay. Now listen the statement carefully. Sir, what we got here, sir? Somatic hybrid. What we got here? Somatic hybrid. Somatic hybrid. Now listen these statements carefully. For example, this is a protoplast. What it is? A protoplast. This is protoplast. Naked protoplast, right? Sir, this is also, right, a naked protoplast. Protoplast. Sir, what is this yellow layer indicates? Plasma membrane. What these dots indicates? Cytoplasm. Cytoplasm. Whereas this yellow dot indicates what, sir? Nucleus. Yellow dot indicates what, sir? Nucleus. Yellow dot indicates nucleus. Is it okay? Now listen this statement carefully. Sir, this is which hybrid, sir? Somatic hybrid. Which hybrid? Somatic hybrid. Sir, this somatic hybrid may be either synkaryon or heterokaryon. What is somatic hybrid may be either what, sir? Synkaryon or heterokaryon. Sir, what is the meaning of synkaryon, sir? Name itself indicates. Sir, syngamy, fusion. Karyon, nucleus. It means fusion of what? Nucleus. Now listen. Sir, this is naked protoplast, this is naked protoplast. With the help of fusogen, we are fusing these two naked protoplast. Yes or no? Fusing the two naked protoplast. It means when these two gets fused, what happened? When these two gets fused, what happened, sir? The cytoplasm of this naked protoplast fused with the cytoplasm of this naked protoplast. It means here fusion of cytoplasm taken place, not taken place, taken place. Similarly, the fusion of these two nuclei also takes place. Fusion of both the nuclei also takes place. Sir, karyon, nucleus, syngamy, fusion. Therefore, here what are fused? Right, nuclei are fused. Therefore, now this is with how many nuclei? Now it is with one nuclei. Is this statement okay? Therefore, synkaryon with how many nuclei? With one nuclei. With one nucleus. With how many nucleus, sir? One nucleus. Who is with one nucleus? Synkaryon is with one nucleus. Synkaryon is with what, sir? One nucleus. Is this statement okay? Clear? Right. But that nucleus is having nuclear genes of this protoplast and nuclear genes of this protoplast also. Therefore, having nuclear genes of both the protoplast. Now, go for heterokaryon. Sir, this is a naked protoplast, this is naked protoplast. Sir, when these two naked protoplasts are fused with one another, what happened means cytoplasm of this fused with cytoplasm of this. It means a fusion of cytoplasm has taken place. Fusion of what has taken place? Fusion of cytoplasm has taken place. Fusion of cytoplasm has taken place. Fusion of cytoplasm has taken place. Is it okay? But nuclear fusion has not taken place. But nuclear fusion has not taken place. Resulting in what, sir? Resulting in a hybrid protoplast having how many nuclei? Two nuclei. Therefore, heterokaryon with two nuclei. Heterokaryon with how many nuclei, sir? With two nuclei. Heterokaryon with two nuclei. Heterokaryon with two nuclei. Is this statement okay? Clear? Right? First somatic hybrid. What? First somatic hybrid was developed by Carlson and et al. Et al means his colleagues. Okay, right. Sir, how this first somatic hybrid was developed, sir? First somatic hybrid was developed by Carlson by crossing Nicotiana, right? Nicotiana gluca with Nicotiana Lance Dorfi. Okay, what is the botanical name of the two plants? Nicotiana gluca with Nicotiana Langsdorfi. right? Sir, these two are species of tobacco. They are species of what, sir? Tobacco. They are species of tobacco. Therefore, when these two are crossed with one another, then first somatic hybrid was developed. It was developed by whom, sir? Carlson and his colleagues. Is this statement okay? Right. Now listen here. Sir, what is the meaning of a hybrid? What is the meaning of hybrid? Sir, hybrid is also called as a cytoplasmic hybrid, sir. Hybrid is also called as what? Cytoplasmic hybrid. Sir, this is naked protoplast. This is a naked protoplast, sir. During fusion, what happened means cytoplasm of this fused with cytoplasm of this. Therefore, what has taken place? Fusion of cytoplasm has taken place. Fusion of cytoplasm has taken place. Now, see this diagram carefully. Sir, fusion of what has taken place? Cytoplasm has taken place. This cytoplasm fused with this cytoplasm. Is it okay? Now, this is nucleus of this and this is nucleus of this. Sir, here how many nuclei are present? 2. Out of 2, 1 disintegrates. Therefore, fusion of cytoplasm followed by what, sir? Disintegration. Followed by disintegration of what? Nucleus. Results in the formation of cytoplasmic hybrid or hybrid. Now you tell me, sir, how hybrid or cytoplasmic hybrid is formed, sir? Sir, it is formed by the fusion of cytoplasm of 2 naked protoplast. After cytoplasm gets fused, out of 2 nuclei, 1 nuclei gets disintegrated. Therefore, hybrid or cytoplasmic hybrid contains, sir, what it contains? Contains cytoplasm of 2 parents, but nucleus of only 1 parent. Sir, why it is having nucleus of only 1 parent, sir? Because the nucleus of another parent has disintegrated, but it contains cytoplasm of both the parents. Having cytoplasm of both, but a nucleus of only 1 parent. That is which hybrid, sir? Hybrid or cytoplasmic hybrid. Applications of somatic hybridization. Applications of somatic hybridization. What are the applications of somatic hybridization? Now listen here. Number 1, first application. Sir, in nature, sexual reproduction is not possible between 2 different species. Sexual reproduction is not possible between 2 different species. Okay, but we can produce a hybrid by crossing 2 different species in protoplast fusion, that is by somatic hybridization. Therefore, a hybrid can be produced. Hybrid can be produced from what, sir? From 2 plant species. 2 plant species which cannot involve in sexual reproduction in nature. Therefore, in nature, sexual reproduction is not possible between 2 plant species. From such 2 plant species, we can produce a hybrid. For example, you take a carrot. What's a carrot? You take rice. Now you tell me, in nature, is sexual reproduction possible between carrot and rice? I am speaking about sexual reproduction in nature. In nature, sexual reproduction is not possible between these two plants. In such cases also, by using somatic hybridization, we can produce a hybrid. We can produce a hybrid. Is this statement okay? Right. Number 2, second example. Take a brinjal. Brinjal. Take tomato. What and what, sir? Brinjal and tomato. Sir, this is one type of plant species. This is another type of plant species. Is a sexual reproduction possible between these two plant species, sir? No. Is a sexual reproduction possible between these two plant species in nature? No. In nature, sexual reproduction is not possible between these two plant species. In this case also, by somatic hybridization, a hybrid can be produced. What can be produced? Hybrid can be produced. What can be produced? Hybrid can be produced. Go for third example. What? Third example. Take a potato and take a tomato. Sir, this is one type of plant species. This is another type of plant species. Right? Is sexual reproduction possible in nature in between these two plant species? No. No possibility of sexual reproduction between these two plant species. In such case also, we can carry out somatic hybridization and we can produce a hybrid. We can produce a hybrid. This hybrid produced by crossing potato and tomato is called as pomato. What it is called as? Pomato. It is called as pomato. Therefore, see, in nature, no possibility of sexual reproduction between carrot and rice. In such case also, by somatic hybridization, hybrid is produced. No possibility of sexual reproduction between brinjal and tomato in nature. In this case also, hybrid is produced by somatic hybridization. Therefore, in nature, even though sexual reproduction not possible between two different plant species, in such cases also, by somatic hybridization, hybrids can be produced. What can be produced? Hybrids can be produced. Right? Thank you.